this talk would be, as I mentioned, focused on the industry challenges in drug substance development. The drug substance, the definition of drug substance here would be crystalline particulate solid in powder form. So usually we are handling white powders, and those are APIs, active pharmaceutical ingredients, which are base for making tablets, solid dosage forms. And then talking about powders, we usually handle crystalline materials. Sometimes they are amorphous, but most of them would be crystalline solids. And then I have identified a number of kind of bullet points related to the challenges. And I know it's not exhaustive list, but those are the maybe the most frequently occurring issues we are handling in our daily routine. So the number one, or one of the most important, would be poor solubility of active ingredients. So nowadays, more and more APIs, molecules, even they are considered small molecules, but they are larger and larger, and they are getting up to even 1,000 grams per mole in molecular weight. And they are also quite flexible, three-dimensional, quite complex. And that actually brings complexities or challenges related to the solubility. So most of those APIs we are handling, they would have poor solubility or low solubility in a range of solvents. And then that would translate to insufficient bioavailability. But from the manufacturing perspective, that could make it difficult to manufacture those APIs with high productivity. So with high yield and throughput. That wouldn't be possible if you wouldn't solve the problem of low solubility. And the other challenge would be, it's a broad topic again, it's polymorphism. Crystalline materials, they can exist in a number of different crystal forms. Those forms, they are interrelated in terms of stability. Some of them are metastable, and then the most stable are not so easy to obtain sometimes. And then we also are getting unwanted solvates or hydrates. So just at the start of the development process, there is usually a polymer screen being done in a range of different solvents, and then many of them solvates and hydrates are being isolated, which is not preferred situation. So polymorphism is another big issue, which is the pharma industry is facing. And then the third one would be, I combined it together actually, the issues related to particle size and particle shape for morphology, because they are kind of connected together. And then the particle size distribution and shape would affect processability of the powder. So filterability, flowability, tabletability, so compressibility is one part of this. Also, particles which would have unwanted characteristics. For example, they would be too large, they would have insufficient dissolution rate and bioavailability. Many often particles are not appearing as primary particles, but they are showing up as kind of granules, agglomerates. And again, this is unwanted because many often this will affect purity of the material. It's difficult to remove impurities which are hidden in the voids in between agglomerate particles. So those are few challenges we are facing. And then in our daily work, we mainly do experimentations. So we do a lot of experiments. We are optimizing processes, but then also we are trying to use computational methods. And I will just briefly highlight those computational approaches we are using. So first I will start from our typical workflow for solvent selection for a crystallization process. Thanks to the I think because we use the computational tools, we are able to screen much more solvents than we could do just focusing only on experimentation. And then usually we start from around 70 solvents, which we screen computationally, and we look for solubility and temperature dependence of solubility, propensity for solvate or oiling, and the capability of impurity purging of those solvents. And then we select less than 10 solvents for experimental validation. And this part saves a lot of time and effort and cost, because if we would like to screen experimentally 70 solvents, you can imagine that would take probably number of weeks, if not months, but we can do it in maybe 2-3 days. And then having those few promising solvents identified, we do experimental validation, so we do experimental solubility. Usually this is HPLC solubility determination. And then we check for crystal form, for purity, and some other parameters. And then we narrow it down to 2, 3, or even one solvent, which is our winner, let's say, in this competition. And then we do small-scale crystallization experiments, and then we scale it up and develop the full process. So, as you can see, the computational part is very important piece of this workflow, and it really works. So, regarding the software we are using, we use mainly two platforms. One is Cosmo Term or Cosmo RS, and the other one is Material Studio. And then with Cosmo, we predict solubility, also propensity for solvate, to crystal, or even salt formation. And then with Material Studio, we do conformational search. So we are searching for a low energy conformation of the molecule, and we use that identified molecule for solubility predictions using Cosmo. So the two softwares, they work together. And then with Material Studio, we also do lattice energy calculations or cohesive energy. We visualize crystal lattices. We are looking for imperfections in the lattice channels, for example, or some other structural features. And also we do crystal shape prediction. And also we started to do crystal structure predictions. And in the future, we'd like to do reverse refinement just to elucidate crystal structures using powder EXR data. So we are just constantly developing those capabilities, trying to do more and more predictive modeling. So starting from solubility predictions, I would like to show some case study when we have an API with molecular weight of around 500 grams per mole, which is not small. And we have a comparison between the related solubility, predicted solubility with Cosmo, and the experimental solubility measured with HPLC. And we have a range of different solvents. And as you can see, there is a good match between an order or a ranking of solubilities. So we can predict relative solubilities. In addition to that, for the same API, we've done the scan or screen for the solvate formation propensity with Cosmo. So for that, we used an algorithm which computes excess enthalpy or enthalpy of mixing. And then this enthalpy of mixing is related to the interaction between solvent and solute, and more negative value indicates more favorable mixing, more exothermic events, and then the positive values indicate unfavorable mixing and endothermic kind of process. And then, as you can see, the red colored bars indicate those solvents where solvates were identified. So those are the predictions. You can see the excess enthalpy values. Try to point out here. But then those are also validated experimentally. So we found those solvates in those solvents. On the other hand, those showed in green, the predicted excess enthalpies are less negative or positive, and we didn't see solvates in those solvents. So that was a very successful validation of this approach. In addition to the relative solubilities, we can also do absolute solubility predictions, and I would like to show this another case study when we compared experimental versus predicted solubility at two different temperatures of 20 and 48. And then, as you can see, for some of the systems, the solubility is predicted very well. But altogether, for most of the systems, it comes into the kind of boundaries of plus minus 20% of error of deviation. And another information you can read from this is that at lower temperature of 20, the predictions are overall better than at higher temperatures. So there are some limitations related to the temperature dependence of solubility. Predictions of higher temperature are not yet as accurate. In addition to the neat solvents, single system solvents, we can also predict solubilities in solvent mixtures. And this is very handy, very useful application because for many APIs, there is no neat solvent which can be identified. Basically, solubility is so low that there's nothing to choose from. And then, in this case, sometimes we are able to find the solvent mixture which satisfies our criteria. And then I'm showing an example of binary solvent mixture on the left-hand side. And here we can see the solubility is predicted for the mixture of ethyl acetate water and at different temperatures. As we can see, there is an increase in solubility for a certain ratio of two solvents, and this is a synergistic effect of solubility, so increase of solubility. And we are usually looking for this phenomenon if we want to find solvent mixture where solubility of API will be satisfied higher. And we can also do ternary or quaternary or higher complexity mixtures as well. So there is a lot of scope and flexibility in this regarding the solvent mixtures. I would like to move to another aspect, which is the conformational aspect in solubility predictions. And I think this might be a little bit more fundamental, and I think it's quite important and interesting on the other hand. So I will show an example of predictions, solubility predictions for malic acid, and in focus on different conformations and just the geometrical features. So for malic acid can make a number of intra-hydrogen bonds, intramolecular hydrogen bonds, and that will be reflected in different related energy. And then there is a hypothesis that for solubility predictions, the lowest energies conformation should be taken into account because this is the conformation which is the most probable or most frequently occurring in solution. But we found that it may not always be the case. And this is the interesting part actually. And then comparing the predicted solubilities, we predicted solubilities in a range of solvents, 5 different solvents, and experimental solubilities are compared with the predicted ones for one conformer And this is the crystal light conformation. So the conformation taken from the crystal, optimized in vacuum with DFT. And another set of predictions is for the conformer set when we use 10 conformers. And as you can see, the values are quite good, even for one conformer they used. They are maybe not perfect, but for us it's already satisfying because we see those differences. From practical point of view, it's not as important to have absolute values, but to have a ranking of solubilities to choose the better candidate than the others. So as you can see, the predictions are quite good, although the approach when we use 10 conformers works slightly better. But it's not always the case that we have such a nice predictions. And when we handle molecules which are larger and which are more flexible, and then the challenge probably kind of increases. And we have another case study we were exploring, which is tolbutamide. And tolbutamide is very interesting from this perspective because it forms intramolecular hydrogen bond, which is quite distinct in terms of energy. There is 5 kilocalories per mole difference, but we've done with molecular dynamic simulations in methanol and in toluene, and we found that in methanol it stays as the conformation is similar to that of the starting conformation used, but then in toluene it turns into intramolecular hydrogen bonded conformation, and that actually our understanding here is that in non-polar solvents or low polarity solvents we should expect more of this intra-hydrogen bonded conformation, and in contrary, in polar solvents we should see more kind of open conformation. And then we've done we generated a range of different conformations, including intra and non-intra hydrogen bonded ones, and we predicted solubilities. But we found that just the hypothesis I mentioned about the conformation energy, the lower the energy, the better conformation and the better predictions, doesn't apply here because for those conformers with the lowest energy, we actually don't see very good predictions. And those lower energy conformations, they are those with intramolecular hydrogen bonding. So there is no clear correlation between the conformer energy and the accuracy of the solubility prediction for tolbutamide. So as you can see, there are some molecules which are more tricky to predict solubilities. We looked also at the electron density isosurfaces, and we found that again there is not clear correlation between predicted solubility and conformational energy of a conformer. But we found that there is some correlation between this location of those polar domains. So we found that those conformations where the domains are kind of connected when we have positive charge put together and then negative charge from 3 oxygen atoms, for example, those give much higher solubilities in polar solvents and low solubility in lower polarity solvents, and vice versa. For those where those polar domains were more scattered or isolated, then we got kind of opposite effects, so low solubility in polar solvents. So altogether, this is something interesting because it shows that there's no correlation with the energy of conformations in terms of kind of accuracy of predictions, but there is some correlation with this landscape of those colorful patches actually here. It would be nice to quantify it somehow and explore it further. And then I would go to the last part, which is the crystal shape and morphology predictions of APIs. And in our projects, mostly we are able to obtain. We are getting a crystal information file, a C file, which is the crystal structure basically of APIs. So we are able to use it to predict morphology of the crystalline particles. And those predictions are considered in vacuum, but still, many often they are reflecting the experimental morphology, which is nice to see. There are some exceptions, but in most cases we see a nice match between the predicted and the experimental morphologies. And just looking at the methodologies, we have chosen an attachment energy theory where the crystal growth is considered to be proportional to the attachment energy, which is the energy released upon interaction with slice, molecular slice of the particular facet. And the higher the attachment energy, the more negative the attachment energy, the fastest the growth perpendicular to that facet. So based on this, we can assess or estimate somehow, we can call it probably intrinsic propensity for crystal growth in particular directions. And also this will give us a theoretical aspect ratio of the particle. But then I will maybe mention that many often the growth into elongated particles is related to higher polarity at the top and bottom of the particles. Basically, more polar atoms are present at the top and bottom, and more polar atoms, they afford more interactions, more hydrogen bonding, and then the growth will be faster in that direction, leading to the elongated particles. But there are some exceptions to this, and we found that another reason when particles are growing into elongated shape can be particular orientation of the molecules within crystal and particular geometry of those molecules. So usually for elongated molecules or those molecules like clofazimine, which is somehow elongated but it's also flat molecule, there is no intermolecular hydrogen bonds, but most of the interactions are those weak high stacking interactions, and even though they are weak interactions, there is so many atoms involved in interactions that the preferred growth is actually leading to elongated particles. This is for form 3, but if we look at the packing of molecules in form 1, where the shape is quite different, it's not elongated, not as elongated, we can see a different packing of molecules. So molecular packing is an important part, aspect or factor which is leading to different shapes. Ideally, we would like to predict influence of solvent on the morphology, on the shape of crystals. And sometimes we're able to analyze in more detail or to find some correlation between the surface termination of surface chemistry of particles and the morphology. Sometimes we are trying to hypothesize actually what could be the impact of a particular solvent type. For example, if we consider polar protic solvents for this particular particle, this particle features kind of high density of polar oxygen atoms, which are coming from carbonyl groups at 001 surface. But then at 100 surface, there is just low polarity the CH2 groups. So if we consider polar protic solvents, they could preferentially interact with those oxygens, forming strong hydrogen bonding, and we expect that they could inhibit growth perpendicular to the 001 direction, and then the particles in polar solvents like alcohols could be thinner On the other hand, they could allow growth more into perpendicular to 1003 because ethanol, methanol, alcohol molecules wouldn't be interacting here too much. And then we found some confirmation of this. This is very preliminary. It's probably not yet fully reliable as we'd like to see, but it shows some trend at least that in ethanol we see more elongated particles. In isopropanol we see the particles kind of less elongated, and then in acetone though they are both lower aspect ratio particles and thicker particles. So this type of analysis we are doing, trying to figure out which solvents to use to achieve the most desired morphology. Usually we want to have as low aspect ratio as possible, as close to cubic as possible. Another aspect in morphology prediction is influence of solvent, which we can study with explicit solvent or implicit solvent. I think explicit is much more difficult, but then in this case we made predictions for implicit solvation models, so we considered different solvent environment, and then we benchmarked it against experimental results. So as we can see, in vacuum we have the most elongated particle predicted. There's no interaction with external environment, and in less polar solvent we can see decrease of aspect ratio. In more polar solvent we can see even more decrease of aspect ratio. So that indicates that there's inhibition of growth at these polar facets of the particle, and that quite nicely matches the experimental results.